Hey listeners, it's Paul Andriola here. Why not join our community at Small Cap Discoveries, where we offer our members direct access to some of the best microcap investment opportunities available. Our members are getting access to premium microcap financings, research reports, and direct access to management. Sign up today at www.smallcapdiscoveries.com.
Hi, everyone. Welcome to the Small Cap Discoveries conference call. Today on our call, we have the CEO, Jennifer Bath from Immunoprecise Antibodies. Immunoprecise trades on the TSX Venture Exchange and also on the NASDAQ, both under the symbol IPA. The company is currently trading at $5.50 Canadian with 25 million shares outstanding, or about a Canadian $136 million market cap. I'd now like to hand it over to Paul Andriola.
Hey, thanks, Trevor. I'm always excited to talk to Jennifer. It's become a lot less common than when I was officially with the company. So I certainly appreciate the time that she's taken out for us. For everyone that's familiar and maybe some that aren't familiar, Immunoprecise, a company that's very near and dear to my heart. And a common discussion point amongst the members of Small Cap Discoveries. But we have, lo and behold, we have Jennifer here with us today, Dr. Jennifer Bath, CEO of Immunoprecise. And Jennifer, first off, very happy to have you here. So thank you for your time. I know you're very busy. But why don't we, before we sort of get into an update, let's refresh everybody's memory and just give everyone that's listening right now sort of a general idea of what Immunoprecise does, and then we'll get into some more details down the road.
Sure. And thanks for having me, Paul. It's great to see you. It's been a while, so fantastic to see you. Great to have this opportunity to provide an update to our investors and to possibly some new people just hearing the IPA story. So basically, the brief side of IPA is we are a contract research organization in terms of where the majority of our current revenue comes from, which basically means we build therapeutic antibody products for biotech and for pharma with a very wide variety and large number of different clients. We've really, in the last year, onboarded quite a number of clients with very varied backgrounds, everywhere from 2 employees that are very hedge fund backed or spinouts of large pharma, through to 80% of the global top pharma. And we're seeing more and more interest in technologies that we worked really hard to bring forward or to build internally, especially over the last 18 months, a few technologies that have really kind of taken front and center in our industry. One being, for instance, what's called a function-first B-cell technology. There really isn't anything out there that is like this technology, and it's really beginning to spread and beginning to get a lot of excitement brewing in the pharma community. With regard to kind of our overall strategy, what we spent a lot of time doing after restructuring this company over the past 4 to 5 years was building end-to-end services. So we could solve some of the biggest challenges that exist in bringing really effective, clinically validated antibodies forward to the clinic for our pharma partners. Starting about 2 years ago, we also spun out an individual company called Talem Therapeutics. It's still owned by IPA, but within there, we build therapeutic antibodies that IPA owns, that Talem owns 100%. And those become opportunities for out-licensing and partnership with various pharma and biotech. And we're really excited to announce in our Q1, which for us actually was just a couple of months ago, our end of our Q1, that we have 4 products, 2 potential best-in-class and 2 potential first-in-class products that are now available for out-licensing. Very exciting T-cell engagers. We have some oncology products in there with a lot of good information, a lot of good solid data behind them that are ready to move into clinical manufacturing. So we're excited about that potential revenue stream for IPA as well. And that's where a lot of our focus on the marketing aspect of our assets, along with the growth in our contract research is. But that being said, probably the most exciting thing that's happening at IPA right now is, now that we have these end-to-end services built, we've gained an excellent reputation for putting products forward that have allowed many of our clients to be successful in the clinic. And they've really come to trust us and the services and the full scope of the services that we offer, which, by the way, we were actually awarded best, most thorough contract research organization in the world just recently by a leading analyst of contract research globally. But now that we have that built, we really, had turned our sights to what we believe this industry will be, what will really transform this industry. What really are the cutting edge differentiators in this industry over the next 2, 3, 4 years. And we have felt very firmly, and I know we talked about this just immediately after our acquisition, our third acquisition of BioStrand last April. We have always felt that will involve machine learning and AI. The challenge that we've seen in our industry is just that as companies have gone out and either organically introduced AI or machine learning, or have gone out and acquired other companies to bring into the mix, there have been a lot of challenges in trying to do that in the therapeutic antibody space. A number of the internally grown AI programs have been very incremental impacts to therapeutic antibody discovery, oftentimes helping just one portion of what is already done in wet lab work, not significantly decreasing the time frame, not really providing insights historically that are really cutting edge or really make a big difference in terms of time and money and energy and quality of the product. And the other, challenge that we've seen is when companies have been purchased and brought internally is that oftentimes a fairly similar scenario. We might be improving some of the candidates that can be screened initially, but they're still going through the whole process of wet lab discovery. We really set our sights, it was actually about 4 years ago, so about a year after I came on, to really get out there, really understand, interview, talk with, meet with, collaborate with, and challenge companies in the AI and machine learning space to figure out, really, where is this going to go? What twists and turns can we expect? What are people doing that we think is not sustainable, is not going to be a game changer? And who out there has something that is unique and differentiating and really can be applied in a way to change how we do things today, from a financial perspective, from a speed perspective, and most of all, from a quality perspective. That's how we landed on BioStrand. So there's a lot that we can talk about with regard to BioStrand, but that's really the most exciting thing that we're doing today. We've been incorporating them into our offerings in the contract research lab. We bring them into existing programs with all sorts of exciting things that we can do to help produce a better product, a safer product, to predict things that might happen in the clinic. And in addition to that, we have spent a lot of time actually coding new aspects to the software, bringing in new capabilities, and creating all sorts of case studies to be able to demonstrate things that we're able to do that have not historically been able, AI and machine learning and software has not been able to do in the personalized medicine space, in increasing the safety and efficacy of drugs, either in the clinic or commercially, and also building these modules to be able to take on very specific markets that we believe we absolutely can transform. Markets like, of course, the personalized medicine space, how we organize and analyze multi-omic data sitting at large pharma companies, how we look at humanization and the use of transgenic animals, both of which really limit diversity and increase and introduce risk into our process. And also the space of biosimilars and biobetters. So we're really busy producing these case studies and beginning to show a select number of companies exactly what we can do to change the way that they do drug discovery.
I'm super excited about BioStrand. I think everybody knows that. But before we really dive into that, why don't we cover off the rest of the business first, and then we'll jump on a bunch of questions I have on BioStrand.
Okay.
The way I see the business, it's almost now three pieces. You may see it a little bit differently, but we get the CRO. We've got Talem, which sort of it sort of takes advantage of all the different other sort of tools that you have. And then I sort of separate BioStrand because it does really feel like it's a completely new and different tool that could be part of your CRO, but it could also stand alone as an offering. So why don't we start with the CRO side? A lot of things have been going on. The revenue component has somewhat slowed down. The last quarter was flat. Can you tell us a little bit about kind of what's going on under the engine right now? Sure. And why did we see flat revenues this last quarter?
Sure. Yeah, I'm happy to. So overall, actually, we continue to see really good year-over-year growth. Quarter one is significantly different within not just IPA, but actually almost all contract research organizations that operate in Europe. So what we typically see in our quarter one, and it's not impacted on a relative basis either from a quarter one to a quarter one, really only on a Q2, Q3 to Q4 relative to Q1, is that we tend not to get a lot of new contract research business in quarter one because primarily in Europe, we have a very shortened period of time for summer where most people have their children out of school and are taking their vacations very seriously. Very, very different than what we see in North America. Very much different than what we see for sure in the United States. Great. And so when it comes to contract research programs where you would typically initiate a program and then stay very in touch with the scientific team working on that program, as we always say, we like to work as an extension of their laboratory. It makes it really difficult for companies, especially companies in Europe, which is where the majority of our revenue comes from, to initiate a project knowing that they're going to not be available for the first portion of the actual program, where we would normally be having weekly calls, presenting updates, and asking them to make go, no-go decisions on the project. So very common for us that we see relatively flat Q1 and that those programs start to pick up in Q2. And that's where we oftentimes see the indicators of kind of where we're going and how we're doing. And we certainly saw that in Q1. I don't have the numbers in front of me here, but I know we had a tremendous lift in the Q1 sales orders. So people who actually went through with getting a quote, they went through with getting a statement of work where that's applicable, and they actually provided the purchase order. So they flip that to what we call a sales order, which means they're in the game for the program, but they don't want to start that program until we actually reach Q2 and they're back into the swing for typically academic calendars related to not vacationing. So that's a lot of what we saw in Q1. It's very common for us. And like I said, in my experience, it's very common for contract research organizations in general. And then we have other indicators. For instance, we do have a pipeline, a weighted pipeline, where we weight the probability of the future conversions from quotes to sales orders. And we also see that up. We saw that up in Q1. We see that going up in Q2. But they're not things that are reflected in revenue. So again, we just have that kind of pause on people actually initiating projects till they're back in their lab and back in their schedule.
It makes sense. Now, the other thing that we seem pretty evident is that, I mean, you obviously have facilities in the Netherlands and the EU, subject to the euro, which has been hit very hard over the last little while. So some of the revenue, call it revenue decline or revenue flatness, is actually a currency translation issue.
That's true.
Is it not? Yeah. Okay.
Yep, that's absolutely true.
Yeah, and roughly how much do you think is impacted by currency?
Again, I don't have that exact number, but I think we did touch on that in the transcript for our day one earnings call. It was several $100,000, but I don't remember exactly, what.
Yeah, if I remember correctly, I think it was 300,000.
That sounds about right, Paul.
Yeah, so that clearly had an impact on revenues. What's the sort of, I guess, the status or maybe even the outlook for you've got a relationship with Pierre Fabre and Eurofins. Maybe touch on a little bit how you see those things going, but more importantly, do you envision other sort of partnerships like that for the CRO side of the business?
Yeah, so that's a great question. So each of those partnerships is a little bit different. Collaborations are going really well. We actually seen quite a bit of cross-pollination of business between Eurofins and IPA. That relationship actually is quite strong. Communication is regular. And again, the kind of sending over of different programs in both directions has been very fruitful. And we've actually just begun an additional area of work with Eurofins. That relates to one of our internal services that we're just preparing to launch. But I think we've actually mentioned on one of our earnings calls, which is our multi-species next generation services, which gives us access to a whole host of very large scale data that this works very nicely in feeding into our AI programs and our iterative AI learning. So a lot more information on what we refer to as our universal fingerprints and ability to do more and more work in silico. But that relationship is going quite well. And, the Eurofins discovery unit we struck that relationship with was relatively new and coming into the therapeutic antibody space and the discovery space, and really a little bit more focused on the downstream. So we're definitely beginning to see the two groups coming together a little bit more forcefully as people begin to get experience in using IPA in connection with Eurofins. Pierre Fabre itself was a research collaboration with Talem to work on primarily oncology targets. It was 9 targets over 3 years, I believe. Those programs are going well. They're underway. Pierre Fabre is not very interested in us discussing too much exactly where those programs are. Which I guess I can certainly understand. But what I think is safe to say is the relationship is very strong. It's very good. We actually just had a face-to-face with Pierre Fabre recently as well, our scientific team with theirs. And they've also expressed interest in expanding beyond just that relationship within the collaboration, but into some, I would say, more significant and interesting areas of collaboration and revenue for IPA. But that relationship is very good. And I think it's one where there's a mutual trust and a mutual interest in continuing to work in the long term together. As far as having other partnerships and different types of collaborations coming forward, we see quite a number of different collaborations brewing within IPA. Most of them involve the interest in our AI, in our in silico work. We started producing some case studies from BioStrand as early as, I want to say, last June, maybe last July. The initial case studies were ones where we were looking at essentially our ability to predict certain adverse events in both clinical products and in commercial products, looking at different products that had failed and different products that had caused either significant adverse events that had resulted in products actually not passing through clinical trials, or adverse events that were so significant that they were actually causing death after the product had been commercialized. The case studies were really a wonderful internal validation for us to see that absolutely the software could have predicted some of these adverse events and fatalities, but even more so predicted with very specific pinpointing to exactly what happened at the molecular level. Like what exactly did that large pharma do? And in the first 2 instances, that we looked at were a Pfizer product and Abbott or AbbVie Bioscience product. What exactly happened at the molecular level that if that, one step or one alteration hadn't been made, could have actually prevented those adverse events. We had very strong evidence to suggest that we could pinpoint that specifically, and that we would have been able to either make slight changes to the product or pre-screen the patients to prevent those fatalities. So that was kind of the first really exciting stuff that came out of BioStrand that we shared initially just on a minimal basis, because first and foremost, we want to share that back to the people who were impacted, by those studies to help them make critical and strategic changes. And then we began to make look at some additional case studies within BioStrand that we took on secondarily because we thought that they would be a little bit more of like an 18-month to 24-month timeline within BioStrand that were kind of our big goals we wanted to address. We came out and we said data analysis, right? Data organization, data analysis. We can organize your multi-omic data. We can help you make sense out of, there's a lot of large pharma companies that collect a lot of data, clinical trial data, various patient data, databases that they tap into from other sources, internal data on their research, but they have difficulty really analyzing all of that data because it's in different formats, it's discordant, they can't do it all on one software. So we knew that was of interest. And we've actually been garnering quite a bit of interest from companies in doing that, organizing their data, ensuring that it's blinded to us and that they can just dial in and use our software to analyze their data. And that's been fantastic. But we knew that was the shorter term impact. We figured we would get interest in that. We have had interest in that. But these other case studies we gave BioStrand, we said that's the big goal with BioStrand. Can we build modules to do X, Y, Z? And within those modules, for example, we wanted to know using those fingerprints, and I'm kind of moving just kind of right into the heart of BioStrand, but using these fingerprints called HIFs. When we look at what they truly fundamentally are at a biological level, these fingerprints are pointing to an activity, right? They're pointing to a function. And that's a basic rule in biology. If you have a sequence, it always equals a structure, and that structure relates to a function. And the reality is that in any sort of life science program where you're looking at a disease, where you're looking at improving something from a biological perspective, all you're looking at is actually the function. Mm-hmm. And so there's a lot of examples we could go into, but at its fundamental core, our prediction was, if that's true, if that holds true, we should be able to take the function of any biological molecule, and we should be able to build off of that to conquer areas like, for instance, humanization of molecules. So when you build any sort of therapeutic, majority of them are made in mice or rats. Sometimes people might use llama for VHHs, or they might use even something a little more unique, like a chicken. That's great, but that's very early stage research. Eventually, you need to humanize those so that they're not toxic when they go into the human body. And there's a lot of risk associated with that, in time and money. Mm-hmm. And one way people have circumvented that is going into the transgenic animal space. But now you have an extreme limitation of the diversity, which in layperson terms, basically means like how many interesting candidates for use in humans are actually going to come out of that. And the reality is it's significantly suppressed compared to when you use starting material that hasn't been genetically engineered. So our first thought was, well, if these fingerprints relate to a function, can we build a module around it that allows us to identify those fingerprints that have a particular function? If there's any order that's required for them, anything in particular that needs to be kept in place to keep them functional. But can we identify those particular fingerprints and then build around it the structure required to keep them active? Mm-hmm. And if we can do that, we absolutely would be able to tackle the humanization market, which is about a $30 billion market. And the transgenic animal market, which there's a little bit of varying information on what the size of that market is, but definitely multi-billion-dollar market. So that was the first challenge we posed over the summer for a case study to BioStrand. Again, thinking that might be an 18 to 24-month window. And they were able to turn around very interesting in silico products within 2 weeks. So that was thrilling for us this summer. It kind of sent us all a little bit into a lot of questioning about, wow, okay, how do we manage this? How do we go about validating it? How many times do we want to do this before we really bring this forward as a potential product for clients? But it was also, so interesting to see that, you could build a product based on structure, not on sequence. That we were able to do this based on maintaining a structure that kept a function. And why that interested us is that the patent space doesn't patent things based on structure. So we immediately realized this was a foray into the biosimilar space.
Yeah.
So if you're, for instance, GSK, and you say, I love Humira, but Humira, say, was still on patent, we can hypothetically, with that software, take the sequence for Humira, feed it into our software, identify the fingerprints, build the structure around the fingerprints that are imperative for keeping the function, and produce an identical functional product that actually doesn't have the same sequence. It varies in structure. And in addition to that, we can actually come up with every potential sequence that would give rise to the structure that would maintain the function. So not only can we potentially, with that software, build biosimilars in order to produce products with very specific functions that have already been demonstrated, but we also could reverse engineer all the possible sequences for you, so that say you are, AbbVie, and you are still sitting on a patented Humira, we could provide all of the potential sequences that would lead to the same structure to enable you to build a portfolio to protect your product.
To protect your patent. Absolutely.
Yes. So there are a number of things we immediately realized this is really what we hoped we would be able to do. And wow, the initial in silico research certainly looks like we are being successful in producing that in silico. Right now, what we're doing is we've gotten that process down from 2 weeks to easily done within a day, with quite a bit of in silico support and research. And we're in the process right now of just kind of iteratively taking what we learned, going through in vitro validation and saying, how could we do this better? How can we do binding and docking studies better to really make sure we're getting good binding, that we are retaining function, to kind of iteratively improve the process. We've been doing that iterative improvement for about 4 weeks now. And we did decide that we would go ahead with our preliminary data and reach out to, quite frankly, I'm going to be honest, I thought it was 3 clients, but it turns out it was 4. To provide, just show them what we did, and go very specifically to companies that we have close relationships with, that are on the larger pharma side, and that we know have projects that have been a thorn in their side, a program that hasn't been able to be completed in an animal model in a traditional laboratory setting, or that poses really complicated biochemical challenges. And with regard to those particular clients, we literally picked up the phone over the course of the next 48 hours and we showed them the in silico data. We said, this is what we're doing. This is what, we believe we can do with this. This is the actual data to show you what we have done. And then from there, we said, we want to try this on program XYZ. And however, we also want you to know this is not a fee-for-service type of work with IPA. You're accessing a technology that is very unique, that where these fingerprints are proprietary to us. We have a large number of patents filed in these fingerprints, which, by the way, as of yesterday, we got very positive feedback on a face-to-face oral review on those patents, where they did actually indicate and suggest that these do be marked for approval. So not the final approval, but very, very tough hurdle to get through. When we were surprised we were asked for a face-to-face on, but it's a complicated topic, and even patent reviewers that specialize in this space really needed to hear it face-to-face. But with regard to that, we said there's a technology access fee, to get access to this technology. We'll build you an in silico profile, and even better, we're going to add on to that our NLP software, which, works on, syntax of language with very specific word boundaries, but more importantly, it uses those fingerprints, which, by and large, when fingerprints are picked up by our software, researchers aren't going to really know what they pertain to, what the function is, right? And then from that, search the literature based on those fingerprints and bring back all sorts of information so that we can provide you with a portfolio of information on your target and on your therapeutic or whatever else out there relates in the literature to that you may not be aware of. So we pitched, we'll prepare this for you, provided you pay a technology access fee to review this, to really get a handle on what we can do for you. And if they want to move it forward, that does require out licensing and then eventually milestones and royalty payments. And we definitely went 4 for 4 on that. All 4 groups were really interested in moving their programs forward and very excited about the case studies that they saw. So that's where a lot of our energy has been focused. We're beginning to share this at conferences. A little bit here and there, we're not opening it up full stream. So we're just sharing a little bit of information on the case studies. And then, beginning to really internally put a lot of our emphasis on how does Talem and how do our CRO, how does the existing data we have help to support, BioStrand and help to walk them through what is really meaningful to these existing clients. As they continue to build on, our initial agreement, which is, let's put our energy into therapeutics and put on hold a little bit, with the exception of one very key client we already had, areas like agriculture or other areas where, their capabilities could contribute. So we definitely have additional partnerships coming down the line. 1 or 2 we anticipate might have a little bit further downstream revenue because it's people bringing programs to us that already exist or already a little more advanced, and they want us to apply all of our capabilities to make them better and make them out licensable. But the majority of which are ones where we anticipate revenue streams quite early after bringing those programs internally.
I mean, you opened up a whole bunch of things that just my mind's blown a little bit here, and I didn't even anticipate some of the stuff that you talked around, sort of patent protection, or the fact that nobody's patenting structure of these things right now, and the capability of going patenting the structure is a massive. I don't even know how to describe it. I mean, you have a head start against so many others, and potentially can. You can go and pick and choose all the structures you want, and that's going to be a massive stumbling block for anybody else who stumbles on that structure later on. Yes. Am I seeing it correctly?
Well, it is. So what people will still have to patent will be the sequence, but the way that we come about the sequence for them is by deriving the structure. So for instance, let me give you an example. So we aren't violating patents because we're using structure, and we also can search with structure. So all of our fingerprints now, in the last couple of months, we used the funding from the Valio grant to actually take the sequences of those fingerprints and structuralize them. So now we have structural hits as well. Those have also been indexed. And just recall that the coding and the indexing of all of these HIPs, both sequence and structural, are what enable us to go ahead and search so rapidly, right? Much like how Google has quite a bit of information indexed. We don't have to search everything. We search using these fingerprints that have been indexed. So when we're actually searching, let me give you an example of one of our very first case studies this summer. We used a sequence that originally came from a chicken, and it had a very specific structure. It had a very specific function, and that function related to not just binding, but functional activity associated with a disease. And what BioStrand did was to take that particular sequence and to identify the HIFs that were required for that structure-function relationship, and then to actually build the structure around those particular fingerprints, and then convert that structure back to a sequence, because you'll still need the sequence to do the patent. But the interesting part was the sequence was now human in nature and nothing at all similar to the sequence of the original chicken antibody. So that's wherein you can go back and file the patent now for an antibody where, ideally you have the same function. You have a very similar structure, but that structure doesn't violate their patent. but you have a completely different sequence that now enables you to patent. And we can reverse engineer. Not only can we go in and build things from a structural perspective, but we can reverse that into every possible sequence to give rise to that structure. So now we build patent protection for you around the antibody with a particular function. And it's really interesting because, going back to what you just mentioned, Paul, about kind of being out ahead of people, with regard to both from a patenting perspective, but also with regard to the software. We're very careful about what we release. And as I mentioned, we did go ahead and file several dozens of patents around these sequence HIFs. We probably will not be doing that around the structural HIFs, just for strategic reasons. But what's really interesting is, the idea of a fingerprint sounds so simple, right? It just sounds so simple, especially like bioinformaticists and different researchers out there might think, oh, we could duplicate this, right? We can figure out what these fingerprints are. And there's only 660 million of them. but the reality is, compared to the amount of sequence information out there that people are trying to analyze, 660 million is small. It's finite. That's why we're able to code it and index it and still make then this software so fast. And there's even fewer when it comes to structural HIFs because multiple sequences can give rise to the same structure. But what people don't realize is that long before the founders, Ingrid Brands and Dirk van Hyfte became, started BioStrand and really began, putting so much time and energy and money into, the development of the optimization of this software. If you go back 10, 15 years, they were already identifying these HIFs, these fingerprints, and it's so much more than fingerprints. So it's not just, these are not just sequences. And intuitively, it would not make sense to a biologist that you could take what the actual fingerprints are and relate that to a structure and a function. And I won't get into that because that starts to get into some very trade secret topics. But what people also would not always realize is that it's not just the fingerprints that matter. When those fingerprints were taken and coded, there were all sorts of things that had to be taken into consideration. For instance, the stringencies and the entropy of what could exist in between two fingerprints. There's not just any sort of sequence that's allowable. And there are certain things that must be present to stabilize and keep those fingerprints functional. So a particular amino acid, for instance, in a particular place. And not all fingerprints can exist in any particular random order. If you were to change a single amino acid in one fingerprint, it will automatically change the fingerprints that can and cannot take place downstream of that. So there is so much interesting physics that go into the identification and the understanding of this. And there's so many things that intuitively people would not be able to just figure out in going through and researching this. And it's really all of the complexities that went into that for 10 or 15 years building into what is now existing that make it also an area where, we keep a pretty tight black box around how we use it, what we do it, and how it's coded. But in addition to that, would make it really difficult for someone to ever replicate what we do.
Right. Good. Okay. So you mentioned that four clients that you're working with that you've presented case studies to. How do you, I guess, what do you expect to be the next step? And then probably more importantly, like if you get a client that says, okay, this looks great, how do you figure out how to monetize it? Like what is this worth, or how do you put a price tag on what you think you can do for these people?
Yeah, so that's a great question. So, the next steps are really the finalization of the agreement, and the agreements by and large, I believe, are all with us at the moment. Each of them is slightly unique and different. And in part because we're dealing with areas that, are significantly different from what we deal with in contract research, most of it around IP, the handling of any information they're giving us as starting material, and the handling of their information once it's internal, and how to protect that and how to keep that separate from any sort of data that we produce internally, because we will keep and protect their data. We won't be trying to learn off of their data per se. We'll keep separate databases. So some of it is within there and just the protection of their IP, the protection of our IP. overall, the format and layout of the financial portion of these seems pretty straightforward. With regard to the actual potential value of the product, since the majority of the milestones are based on the ability to progress with that product, then that kind of dictates, how much actually gets sent back to BioStrand and or IPA with regard to the monetary value, as it just depends on the success of that. When we get to the royalty area, in each of those cases, we've been discussing a percentage. And so again, that just really kind of lends back to the success of the product, which leads directly into, the patient need and the market value for that particular product. But in all cases, that portion has stayed relatively similar, with the exception of one group that is providing a little bit more information upfront, actually some sequence data upfront that will be used in the course of optimizing their program. And just for some particular securities around the IP of what's coming in, they'll actually be doing the AI portion also as a fee for service, but that doesn't eliminate the technology access fee and upfront payments and milestones, et cetera.
Okay, so
but it's relatively straightforward from that perspective. It's more of the contractual work is a little bit different.
Right. So it sounds similar to sort of the old BioBuck type of deal where there's some form of upfront payment and then milestone payments and royalty if it becomes successful. That's kind of what we can expect. Okay,
exactly.
Perfect.
Perfect. Yes, and that I think was fully expected by these groups because we didn't see any reservations at all, and it just really relates back to the ability to access that technology.
Wow. And as of yet, it's unproven, right? I can just imagine that if you can make a strong sort of statement by validating this through success, I can just imagine that the upfront fees and everything else become a little bit more expensive down the road.
So yes, that is true. So right now it's kind of interesting. We're in the process of wet lab validation of some of our initial products. And we actually at this point have been producing products more quickly than we can actually get them synthesized and analyzed. We're also producing a variety of different types of products. And so some of the products it will be kind of interesting in terms of us validating them because they're a little bit more sophisticated than a traditional product in some cases. And then in addition, though, to our wet lab validation, which we're fully expecting, to have done in the relatively near future here, the wet lab validation will be looking at binding, strength of binding, and also in several cases, function. But in addition to that, with these initial relationships, collaborations, we have also asked for the right to be able to turn around and use these as case studies. We'll blind, they're very sensitive data, of course. But for us, it was really important that if we accepted them as an early adopter, that they helped us to, bring these capabilities forward. And then I think, our biggest challenge after that will be, do we keep this traditional platform? Can we keep up with that? From a capacity perspective, like I said, they're getting faster and faster every day and turning out these new molecules. But, with regard to that too, is it, there might be some potential to go for some larger contracts up with financial payments up front to just manage, for instance, 10 programs for this company that was initially, an early adopter. And that's something that we are also bringing into the conversation with them. That they'll have the right to, have, some kind of early dibs, if you will, as coming in for a larger program early adopter, so that they don't face capacity issues when and if this is actually the success that we believe it will be.
Gotcha. So to be clear, I heard you say that it sounds like sort of even prior to some sort of finalizing of agreements, you're already taking the steps of sort of validating in the wet lab some of the data that you're finding. Is that correct?
That is correct.
Yeah, fantastic.
Yep.
That's fantastic. So so it sounds like you're fast tracking everything. I can imagine returns or results should be relatively imminent once everything's signed off. So that sounds fantastic. So, I mean, clearly you're excited about BioStrand because we were going to save that for the end. We barely talked about Talum. I do want to get back to BioStrand, but almost now it's almost embarrassing to say this, but it sounds like Talum is kind of the second horse or the second favorite child here. Talum. So you did touch on the 4 products that are in sort of the licensing stage. Like, I mean, there's been a lot of discussions around timing and things like that. How long did these things usually take? Give us a sense. Like, I think there's a preconceived notion that you come up with an asset and all of a sudden you make 5 phone calls and somebody turns around and is interested in buying it. That's not the case, right?
No.
There's a lot of back and forth. There's a lot of biobuck deals we see are based on companies that have had an existing relationship. So these things don't happen overnight. They do take time. Maybe explain a little bit more about sort of the timing and if anything, what are you guys doing different at Talum now that you've got these assets? How do you go and showcase them to the world and try to license them?
Yeah, so all very good questions. So first, yes, with regard to getting an asset out there with regard to how long it takes, what that process is, it's actually not uncommon for discussions around product out licensing to take up to a year. So that's something that's just kind of relatively well known in the industry. With the existing assets we have a couple of different options for groups if they, for instance, are interested in seeing. One of the things we've done to date is we've worked very closely, for instance, on these 4 assets with a number of larger pharma and biotech who have said, hey, we'd love to see this product, for instance, as an ADC carrying a payload. How does it work? Or with our payload? Or we'd love to see this particular asset as a bispecific. And we've actually been going back to the drawing table, working with those groups, getting proof of concept data, kind of going back and forth in this iterative conversation with interested parties where we've been continuing to make investment in bringing data forward for these clients. And in some cases, there might still be some justification or good reason for doing that, in particular on, for instance, a bispecific we've produced that has really garnered a lot of interest. But in other cases, in addition to that, this is kind of pre-actual, hard negotiations on paper. There was also a point where we say, all right, these assets are really of interest. There is good data. Now we're going to draw the line. And those initial conversations, by and large, for us, were happening with people who develop therapeutics within various biopharma and biotech companies. They're the scientists that really enjoy having those conversations about what would be ideal, what would be optimal. But back to your other question about, who do you talk to? How do you get these deals done? That is a very different group of individuals. Those are head of, like corporate operations. These are head of business development. These are head of in-licensing. Depending on the size of the company, they're going to have different, titles. They're responsible for making the business decision. They're the ones that have the control over the budget to make the decision, bring those products in. And so, with those 4 products, the way those got pinpointed was we've done that iterative back and forth, with these discussions at different scientific conferences. We're ready to move these forward. We will now at this point draw the line. Again, there might be a good reason for one of those to do a little more work, but draw the line with regard to what we're going to continue to put forward, because anyone who knows scientists will love to keep working on those for years and get every piece of data out them. But then there is the business side where we say this is ready to go. And the products actually can be priced based on how much work has been done up to point. If that person wanted, for instance, animal data on one of the assets that doesn't have animal data, there can be an adjustment into where, some of that upfront money goes into the milestone payment for when they conduct that in vivo data. It de-risks things for them, still helps us to commercialize the product a little earlier, but also, to reap the rewards at the higher value inflection point when that work is done. So there are conversations right now of really shift to having the conversations with the now primary decision makers, less so with the actual scientific audience or the people who would normally kind of look at replacing a program of theirs internally with that and going directly to the people responsible for filling their pipeline. And we've been engaged, and for people watching us on social media, you'll see we're being engaged at very different types of conferences now, not the larger, broader conferences we're exhibiting at. We still do that for contract research, but you're seeing these smaller, more specialized conferences around topics where we're bringing products that we believe, again, are, I say, for instance, a first-in-class oncology product to an oncology specific partnering meeting where those different companies are sending people with the authority and with the capability of making a decision on a particular product. And with their primary goal is there to sit down one-on-one with people, review the details, less so the big scientific deep dive. We've now moved those into databases, but more so, the general market for these products, why we designed them a certain way and what the potential is and what our pricing, early stage negotiations will be. And so that's part why you'll see kind of an uptick in the conferences, but also very different types of conferences that we are now at and attending for those. And so Talem definitely hasn't taken a back burner, but there's been some shifts. We're really interested in moving some of those products out. Talem is now also pretty engaged with BioStrand. And in some cases, for instance, the case study example I gave you where BioStrand took an antibody that was actually formed in chicken, which is becoming, wild type chicken is becoming more and more popular for therapeutic discovery because it's unique. It's much different than the human antibody repertoire. So it's able to see a lot more, if you will, like see different places on an antigen to bind to. It gives us more diversity. And, we have some programs run in chicken that would need to be humanized eventually. And so we use those in Talem, to go back to BioStrand and say, let's see if we can do this, right? This is a common problem people have, but it was also something that, Talem would have needed to do to bring those products to the human state prior to, moving them into the clinic. And so, which in this case would have been done by a partner. And so there are some really great collaborations that have formed between BioStrand and Talem that we also would like to emphasize and to be able to focus on. And then, of course, that whole area of, our version of a biosimilar, because it doesn't really also need to be something that is, off patent. Is also an area where we want to make sure that Talem has the opportunity to collaborate with BioStrand and move some really, really interesting programs forward. So we've got a lot going on, certainly in Talem. But first and foremost, our emphasis over the next 2 quarters is commercializing those assets that we really believe are these first-in-class, best-in-class products that we know have garnered a lot of interest, that we have had a lot of input on. We know we have some interested parties in, and now really to find them a home so that we can focus on integrating our BioStrand capabilities more forcefully.
Jennifer, when it comes to licensing specifically these products, do you feel you have the bench strength or the sort of the right people that you need to negotiate these things or to push them through? What can you tell us about that piece there?
Yeah, that's a great question. So we have internal support for business development. We do not have internally on our payroll kind of the hard-hitting negotiator with the experience that we would like to see. It did not exist in our BD department, and it was something that we had hoped that we would be able to grow and support through education with prior personnel. What we ended up doing was supporting a newer individual within the company to really be able to support the approach, the materials, the product readiness. And then actually we have brought in 2 external groups to assist us over the course of the last 3 to 4 weeks. In particular, one individual that we've worked with very closely for several years, and that I myself have worked with very closely for about 10 years, who basically did this for large pharma for almost his whole life. So someone I know and I trust, and someone who has a very, very successful history of doing this for a very well-known pharmaceutical company. So starting about 6 weeks ago, probably, we began getting feedback on what's being presented, how it's being presented, the process, the conferences, who we're approaching, et cetera, and even pricing, layout of the structure. And then more recently, I've set our focus on actually, having outside external individuals involved in initiating those conversations and bringing different groups to the table, but also overseeing what is being presented and in what order and how, and then helping, they're on standby to help close negotiations. Until we can find that same type of person internally at IPA. That's our current approach. So far, I actually think those people with that type of experience and that type of history are really hard to come by with the type of success rate that this individual has. And so I really think that's the right path for IPA with where we are right now. And then we also are in the kind of last stages of bringing on a group that we've really been negotiating, because we don't want to make any sort of payment to a group unless they're successful, right? And that's not the normal business plan, but we believe firmly in our products and they will be successful. And if they do too, then we're happy to bring them in on a finder's fee or success fee basis. So we did spend a considerable amount of time looking for a partner that could also assist with that on a broader scale, because we obviously do have a number of products kind of in that boat. And so we're looking at, and likely will bring on one additional group that really is kind of backloaded, like a couple percent on a final finder's fee for a product. Being out licensed. So that's our main focus right now, because by and large, no, I would say that, we haven't found that heavy hitting, really successful person. And even though we have reviewed numerous applications, that just has that experience. We have a wonderful, wonderful BD director who's fantastic. But when we really look at that kind of global multi-decade experience in closing these hard-hitting strategic deals, I think the path we're on is going to be the right path for us.
So what I just heard is you don't have that person internally, but you've hired an outside consultant that basically has the skill set that you
absolutely
wish you had internally. That's what you're saying. Yeah,
exactly. Yeah,
perfect. Awesome. Okay, well that sort of covers those assets, but the last asset we haven't spoken about, and sort of the big one, and the one that certainly is, cost a fair bit of money this last quarter. But Polytope, where are we? What's going on? What can you tell us about Polytope?
Yeah. So, what's going on? So the product itself is through production by and large. We're waiting on the paperwork to catch up with the actual final products that will be ultimately our drug product that gets distributed to the clinic for the clinical trial.
So to be clear on that, because there's a lot of sort of inexperienced life science listeners and people here.
Sure.
When you say manufacture, I mean you're basically making the drug right now, right? You're making
we're making the final product that goes into the human.
Exactly,
that's right.
Yep. So we're not just making some stuff, we're making a drug.
We're making the final drug, the one that actually goes to the clinic. That's the actual one that gets injected into the patient. You bet.
Yeah.
So it is
and it's going to meet all the high standards that, like all the manufacturing standards, all the what is it, GM?
Yeah, it's under GMP conditions.
Exactly.
It's what we call ISO classified suites. So these are specifically rooms with very, very specific protocols for any sort of product that's being produced that's going to go into a human. We're at a facility that has been audited by the FDA and by the EMA to ensure that the product that they're producing, the products that they're producing are ones that are in environments that they would, both regulatory bodies would be comfortable going into humans. And yes, the protocols are, they're very strict protocols, obviously, with regard to pretty much everything that happens, but in particular, traceability, exactly who touched the product at what point, what were the environmental conditions in the room, who made every reagent that went into it. It involves making the final drug product. It involves making sure that that product is stable so that its shelf life at the clinic will be one that we understand. We know what the expiry date should be, and at what point we wouldn't feel comfortable putting an expired product into a patient. It involves the final formulation of what it is actually mixed with, for instance, saline, prior to going into the patient. It involves the vialing in the final product, that goes to the clinic to administer the product to the patient. So those are all the things that are underway right now. Everything that's required for regulatory review of the product is completed. There is one, and we should be right about this point right now, one check that they always do again on the stability so that they can determine the expiration of the product. Typically not a problem with something as traditional as what we have, an IgG1 antibody molecule. And then that's the final information that goes into the submission. Many of you probably heard us say that we actually are leaning toward that submission at the EMA with a clinical trial application as opposed to an IND at the FDA. The rationale for that is we got good feedback from both groups. FDA was quite happy with the product. There were some things, they have a little bit more hoops, we would say maybe a little bit more focused on the safety side than the efficacy. There's always got to be a balance. And we're interested in seeing the individual products in human as well as the whole kind of combination therapy, if you will. But no concerns. They didn't raise any red flags. They actually were the ones who sent us over to the NIH and connected us with the NIAID at the NIH recently, saying that, your product has sustained all of these variants. We want to see the potency and the efficacy of your product compared to the existing commercial products and the other products that are in the clinic, especially as we saw some of our very last candidates that were commercialized drop out when Omicron came along. And that's when we got the notification that they'd like the NIH to take over our analyses. And they now are doing that. We're actually backstocking product with the NIH so that as soon as a new variant emerges, they can pull out our product, they can test it, they can get that done very rapidly. And now the FDA has access to that to see, who's still alive. Normally they just do that for, clinical and commercial products, but it was really the history of sustainable efficacy for ours that caused them to bring us into that same sort of routine with the NIH. So that's been a great relationship with us. We've always had good feedback. We've always had positive feedback. Didn't have any red flags in the process of preparing our work that's gone to review for them for the IND. But what was really interesting is we decided simultaneously to talk to the EMA. We thought, let's get their feedback as well. Let's see how they feel about this product, because wherever we can see a gain in time or a gain in benefit for the company, we'll want to take advantage of that. So we send them the data as well. And that was very interesting. They also, really appreciated the product. There were no red flags. They loved all the data that we had done for the CTA application. But they also gave us feedback that, one thing we don't talk a lot about, but we did early on, was we have the ability to plug and play with our antibodies very strategically placed in different positions on this virus. The idea was not only that we wouldn't drop off and lose efficacy, but we might also be able to, one, prevent additional viral replication when mutations do occur, because we have synergy where the antibodies help really the overall efficacy to be increased. And we might also be able to stop then that viral replication to prevent any additional mutations from arising in patients in the virus within the patient when an existing therapy is no longer as efficacious. So that plug and play aspect was really important for the overall eventually sustainability, even though right now to date, again, we're the only first generation therapeutic that is still efficacious across, with also a very short range of change in efficacy across all of the variants. We haven't needed to plug and play yet, but we've always felt it's important to have those in the background, right? So what we did over the last year and a half is we engineered and optimized all of these different versions of the antibodies that we currently use. So we can shift a little bit this way on the virus, we can shift a little bit that way, we can bind a little stronger, we can change depending if at some point there's something that causes us to drop in efficacy. we can modify very rapidly. So what was very interesting about the EMA was they said, not only do we love this, and yes, absolutely, this looks good to us, but we would allow you to swap in those plug and play antibodies anytime during the clinical trial if you wanted. If, for instance, what hits us this winter is really challenging and there's a mutation over here that is near one of your antibodies, and you're interested in plugging in a different antibody to see the efficacy of that during the clinical trial, they would not see an issue with that. And they did not want to see individual antibodies tested in different patients. They're fine with all of them going in simultaneously. So that was also of great interest because that significantly decreased the cost of a phase 1 clinical trial. And then in addition to that, they had an interest in us expediting phase 2 through phase 3 with regard to being able to get that product through more rapidly. And they suggested that without us asking that in their feedback. So for a number of different reasons, we were quite interested in pursuing the CTA application with the EMA. We are in the process right now of that phase 1 clinical trial design. And right now we're leaning toward running that clinical trial in Turkey. But I think, one thing everybody always needs to stay aware of is right now, while it's a bit odd, we're not quite entirely sure why a lot of the reporting is going this direction, but a lot of people feel quite safe with regard to COVID. They feel that it's kind of turned into something that's maybe not as threatening. We absolutely don't see it that way. We see that infection numbers are still high. We know that mutations are still occurring. We know that even right now, we're in the midst of a new variant beginning to rapidly take over in prevalence. So our eye is really on what happens in the fall and in the winter when, we would logically expect to be able to see additional mutations. And because of that, we need to be mindful too that certain places might be right for clinical trials. Certain places might have much higher infection rates and infection burden. So we're preparing to be able to pivot the location based on any information and intel that might really help us to get as much data on endpoint and patient enrollment if needed. But that doesn't change per se our cost or our timeline. It just means that we want to make sure that we hit the right place and time it with the right time with regard to what happens with COVID this winter.
Yeah. And to make it clear, the EMA is the European, I think it's European Medical Association, is that correct?
Yeah, that's exactly right. And a product approved by the EMA, also there's reciprocity. So we're able to market that product in North America as well. So there's no loss of potential market by using EMA instead of the FDA.
That's fantastic. Now, I get pushback every now and then saying it's taken so long, but remind everybody, how long does it usually take to get a therapeutic, especially an antibody therapeutic, sort of from discovery to commercialization?
Yeah, so normally significantly longer than we have done, about twice as long. And in addition to that, and that would be when all of your energy and effort is on that, right? Outside of that, it can take significantly longer than that. A couple of other things for people to remember. There was, kind of a question mark around whether you needed to have a combination therapy to retain efficacy around SARS-CoV-2. And I always kind of laugh about that a little bit when we look back, when we started working on this therapy back in January of 2020. Not only was IPA it's so much in its infancy of its restructuring and building our capabilities, but so really firmly resolved in those capabilities and in our scientific rigor and our ability to tackle this, that, when we came forward, we had very little money. We were not able to spread money across multiple different avenues and go with the one that worked best and run things in parallel and in advance begin to build, some of the things that we might need later, but might not have needed later. But we really also tried to educate people about the virus, about RNA viruses, about what we expected to see with this virus, so that we could also help people understand why a combination therapy was so important to being able to get ahead of the virus, to not chase the virus with something to be efficacious, but to get ahead of the virus, to have something that would retain sustainable efficacy out there in the field. Also something that could help decrease the replication and the number of mutations that we would be dealing with. You know, we talked about these things. We talked about the emergence of variants and how we saw this playing out. And we got a lot of resistance in that first year. I would say really all the way up through about January of that next year, we received just a lot of, you know, interesting feedback. You can even see some conversations taking place on LinkedIn where we went ahead and engaged people and talked to people about how we saw this, you know, playing out and why this was so important. And it was very interesting because, you know, you had a lot of groups that were in the space of therapeutic antibodies, and we started seeing them dropping off right pretty early on, right? We saw some come out where we also were a little bit vocal and saying that one's not going to last based on the epitope, based on, you know, what we're seeing here. There were a number of times that we saw that some had dropped out because we run benchmark studies in our laboratory. We're studying everybody else's alongside ours. So there were times when we knew other ones had fallen out, and yet those companies had not come forward to talk about that, to discuss it in the public realm for quite some time after. But in addition to that, you know, as we were moving these programs forward, we saw different groups trying to kind of recover what they had put forward. If they had two antibodies, whether those were against different parts of the virus or not, one would drop out, and they would try to find something immediately that would jump right in where theirs was, so they didn't have to, you know, slow down to bring something through the system. Which is it's a great way to not scare your investors, but it's not a great way to strategically plan for a molecule that's going to retain efficacy. And sure, lo and behold, even, the companies that put the most money into, and maybe were the most vocal about, bringing products forward, failed to retain efficacy. And so, it's really interesting at this point where we are, and is not the fastest route, in part because we didn't have all the money that our competitors had. We didn't have the lobby groups working to get the federal money. The Canadians thought we were American, and the Americans thought we were Canadian. We had this as well. But in addition to that, it took a while even for regulatory bodies to realize that a combination therapy was really going to be the only way we were going to see sustained efficacy. And so, you look back now to the difference between what the FDA and the EMA required of us. We jumped through a lot of preclinical hurdles trying to convince the FDA that absolutely you need all four of these antibodies. There are things that we don't discuss about why you need all four, because they're involving the synergy. We don't get into all the scientific details, but there are some in there that most companies would have thrown out and thought you don't need, and you absolutely do need them. When you combine these antibodies, you get things you don't see anywhere else based on that synergy. And we have mechanisms of action in place that really have not been considered in most therapeutic antibodies. And so it really did, the FDA wanted us to show you absolutely have to have all four. You can't just use these two. You can't, and things that I think once by and large the world realized we were losing the race with therapeutics. They were dropping out. We didn't any longer have anything available to give people in dire straits, that actually this was the right path to go forward. And I think that's why, EMA got so excited when they saw, the combination therapy we brought forward. So, yes, we took on a bigger challenge than what most groups took on. We did a lot of research behind the scenes. We saw things move forward with, antibodies retrieved from a single patient from what's called an expanded B cell repertoire, which also means minimal diversity based on the process in which they're brought forward. And is it a faster way to do it? Absolutely. Is it worth staking the long-term reputation of your name for scientific quality on? We said no, it's not, and that's not our platform. And we didn't have advanced funding, from to set up a platform like that. We didn't have advanced funding for pandemic preparedness, but we really felt using it to showcase our rigor, to showcase the quality of our products, to showcase that we're serious about bringing forward things that are safe and efficacious. Also, as much as saving lives is important when it comes to COVID, it was also important for us to be able to show what we're capable of doing, what we can do for our partners and for pharma, and that you know, the power of the technologies that were used to bring this to the table. So yeah, preparing a combination therapy is much more time consuming. Preparing it in sequential order and not in parallel when you don't have $200 million to put toward it takes longer. Ultimately, do we believe that this will be a successful commercialization and that this will retain efficacy and will be a hit for IPA? Absolutely, we do. We stand behind that, or we wouldn't still be moving the product forward. So yes, I think it is difficult, you know, we know it's difficult from the outside looking in. We absolutely know that, but we do know that every day there are things being moved at the government level, you know, at the product level. And you know, honestly, December, January is going to come fairly quickly for us to start getting that first in human data.
Yeah, I remember, I think you mentioned to me, like prior to COVID, there'd only been one cocktail antibody therapeutic ever. And I think that was for Ebola, if I remember correctly. So outside of that, we've seen a number of cocktails sort of, you know, show up and prove that it's a viable method of therapeutics. And you know, clearly, I mean, I've got to say it, and I've said it so many times, you said so many things early on about what was likely to work and what wasn't likely to work. And I can't believe how accurate you really were. So hats off to you for sort of sticking that way. Okay, so Polytope itself, I mean, we saw a big chunk of money go out this past quarter. Can you give us a sense of how much more money you think you're going to have to spend on Polytope to sort of get it to where, I know, I mean, the goal is to sort of hand it off, right? You're not going to take this to phase 3 studies.
No.
How much more do you think we end up spending to get it to where we need to get it?
Yeah, no, that is a great question. So, I mean, right now we'll definitely see a tapering off of the expenses. The majority of the manufacturing expenses hit at one time, which was last quarter. And so we had budgeted actually more for that. So it was not a surprise to us. It wasn't anything that was unanticipated internally. And it doesn't change our perspective on our, you know, runway here with regard to cash. But we knew that would be, that's a surprise to some people with the manufacturing process, despite the fact that it's actually tremendously inexpensive, the deal that we have with this particular group. We didn't give up any equity. They believed in the product. They brought it in. We're paying significant, let's just say this way, because there's a very big range of what you can pay, and not also without cutting corners and introducing risk in the quality of the product. There was a company that was actually attempting to get us to actually buy one of their COVID products. They wanted us to fold it into our cocktail. And we had to explain that, you know what, from very early on, we had 97 neutralizing antibodies to choose from. Very, very different than some of our competitors that ended up with 1 or 2. We were very strategic about which ones we took and exactly where they bind, and did all those tests on synergy, right? There was a lot that went into the compilation of that. So we're really happy to test your antibody, happy to give you some information on that, happy to see if it does anything that surprises us, but probability is that no, we're not going to bring that in. And they thought, but if it was stronger and if it actually reduced, you know, the amount of products you had needed to manufacture, that could be a bonus, right? So we exchanged notes on what each group was paying for the manufacturing of their antibody. And we are paying overall to manufacture all 4 products in parallel. So there's no time delay in our actual manufacturing process, less than what they paid for their 1 antibody to be done through GMP. And then, of course, that single antibody of theirs dropped off. Right. The deal that we struck is extremely, extremely inexpensive compared to most. And like I said, now you've seen the majority of that come through in Q1. So we'll definitely see a drop off here. The majority of the remaining fee associated for this is the final vialing and formulation of the product as it goes to the clinic. And then the clinical trial fees. I think before I go into any specific numbers on the clinical trial fees, I'll probably reserve that for us to go. It's not that I don't want to share that. It's just that I need to make sure that I have the exact final numbers on that. It's phase 1 and phase 2A together are what are being calculated for. But there's some of the intricacies and dependencies on that in terms of it's outsourced to a contract research organization in Europe that is doing that clinical trial design. And the final design parameters around the patient enrollment and the location will definitely can have an impact on that. We are prepared for that. It is probably not as expensive as most people believe it could be. We definitely are prepared for that. And in addition to that, you are correct. We're not planning to take this through phase 2B or phase 3. We have made accommodations for enough product to be made for those, so that if a group wants to continue on smoothly and seamlessly into those phases without having to go back to manufacturing, they can do so. While at the same point, they can also just transfer the master cell bank internally if they have commercial capabilities to seamlessly move right into commercialization, which is beneficial for us too. We want that to move as quickly as possible because, of course, we'll have financial milestones and royalties attached to phase 3 and to the commercial product as well. So we've done everything that's necessary to make sure that that is streamlined to very rapidly be transferred to a pharma partner. And the individual that I mentioned to you previously that we have this long-term relationship with, who has had so many successful negotiations in this area, first and foremost has this priority on PolyTope and is very, very familiar with our program, has been clued in and connected to that program for a significant amount of time. So we're still confident that with just early safety and toxicity data that we'll find the right partner to continue forward with that program.
So, and just to make it clear for everybody, so phase 1 and 2A, what do you end up finding out in those two phases?
Yeah, so phase 1 and 2A is typically around the safety of the product. Is there any sort of toxicity or adverse events that they're seeing as the product's being utilized in patients? That's first and foremost, what the point of a phase 1 trial is, whereas phase 2A starts to move into efficacy with specific endpoints. Now that being said, obviously product is going into patients in phase 1, so you do see endpoint data on efficacy. It's just for the sake of how we describe a phase 1 trial, that's not really the purpose of the phase 1 trial. But we do see efficacy in all of the data that comes forward. And what we're really looking for, in addition to safety and toxicity, is the ability to reduce the spread of the virus. We're also looking for the ability to reduce symptoms and reduce hospitalization, and exactly which endpoints get measured and which endpoints get put out for the EMA to analyze, to make their decision on, will be a final decision of our clinical committee in the final design of the trial.
Okay, fantastic. Okay, enough about PolyTope, enough about Talem. We'll take maybe spend a few more minutes on BioStrand. But before we jump on that, I've got a couple questions that have come in. Hopefully it's okay that I ask a few questions here. Let's go right up here. Thanks, Dr. Bath and IPA team, for your efforts. I need to know your thoughts about demands of personalized medicine drug development. I think it's a bit of a generic question, but maybe give us a few seconds on your thoughts on what's happening.
A few seconds on personalized medicine.
Give us your 20 plus years of experience in about 30 seconds. Yeah.
It is. I personally, I am so excited about the personalized medicine space with our capabilities with BioStrand's multi-omic data analysis software. The demand for personalized medicine is huge. It is about a $5.4 trillion market in personalized medicine. There are a lot of areas being researched for personalized medicine. There are a lot of companies out there whose names you will know as everyday names that don't always say it, but where their first and foremost focus is in personalized medicine. But I think the one area that we can all really relate to the most, either because of our personal experiences or the personal experiences of people we know and love, is the impact personalized medicine could have on oncology. And I know I mentioned this briefly. I'm not sure if it was with your group, with SCD, Paul, but You know, right now, for the vast majority of cancers, when people are diagnosed, you know, it is really more about what organ the cancer is in, or what tissue, or maybe it's a, you know, a leukemia, more of a bloodborne cancer. But the prescribed treatment typically for dealing with cancer is to what we call cut, burn, and poisons, right? So we try to remove whatever we can, we try to radiation, and then we go for, you know, whichever we'll just use the FDA as the example, whatever FDA approved drug has been approved to treat that type of cancer. And by and large, unless it's one where we know there is a defined biomarker, it is relating to, you know, whatever drug was used in a clinical trial, which might have only been efficacious in, say, 35 to 40% of the patients in the trial. That would be enough by FDA standards to get that drug approved. Now, whether it's going to work for you is a 35 to 40% chance. It's really, if that patient population in the clinical trial was representative of a standard patient population, that's your probability for success. The beauty of personalized medicine is that what we're saying is that, you know, we need to understand what your cancer is, not where it is, what it is, like what happened. And that's not just a genetic mutation. People need to understand it's not just a mutation in your genome. Sometimes there's a mutation in a gene that actually never shows up in a mutation in a protein. So let's just go back to that basic premise of sequence, structure, function, right? If the sequence doesn't impact, if the mutation in the sequence doesn't impact the structure, it's not going to impact the function. When you have a disease, any disease, cancer or any other disease, what's really happening is a protein isn't functioning properly. That is the bottom line. What we need to do is to find drugs that match the change in the structure of the protein. That is really what personalized medicine is about. So right now, you know, the biggest demand is, can we better treat cancer so that we are not providing people with drugs that don't have a high safety profile, that are attacking all of their other cells, that are causing all of the side effects, and sometimes actually, you know, leading to their death instead of the cancer itself, and not have to provide, you know, radiation where we significantly immunocompromise the patient. And the only way to do that is really understand what mutation has occurred and what structural and functional changes it's caused to the protein. And so, you know, I've talked a little bit more recently about the fact that we're able to do a genomic analysis on people. Now we're able to do that really overnight for as little as $100, depending on the company that you go to. But no one's really been able to adequately assess what that data means. And we're not able to tie that in from genomic data from a patient to transcriptome data. So is that gene being transcribed? Is it being converted to RNA? And is it being converted to protein that has a structural malformation? There's no piece of software in the world that can take all that data and analyze it, except for Lens AI with BioStrand. And so, you know, when a doctor can send out for, you know, your genomic sequence when you come in with a particular form of cancer, but he's got to be able, or he or she has to be able to analyze that and then make something meaningful out of it. And that's where we believe that our software can make a huge difference. And the NLP portion of bringing in everything that's known out there simultaneously, parallel, and immediately with regard to what's known about the fingerprint, which is the function. And so the demand for that is obviously huge. We haven't made huge strides in our ability to solve most cancers, to be able to treat patients more effectively. And it's I think by and large, mostly universally believe that personalized medicine is the best way to get there. Not only do we think, just real quickly here, that our software is a game changer in our ability to analyze patient data, but it is a game changer in the ability to analyze mass amounts of clinical trial data to better understand what are all of the mutations that are out there giving rise to these diseases. What patient population do they represent? Are we getting the full picture of the different mutations that might come forward, and what, for instance, like different ethnicities might be more impacted by different mutations? And then in addition to that, you know, being able to analyze all of these different types of data to understand what to target to make new drugs, which is a huge part of where we see this software coming in for building the new drugs. And it informs us on what drugs to develop, what is the likely size of the patient population we can help with this new drug, so that when you do analyze patient data, we're developing the right drugs to put together with the piece of the puzzle. And these drugs aren't based on where your cancer is located. They're based on the exact structural and functional change that happened in your body that gave rise to the disease. There's huge demand for that.
Yeah, I mean, the analogy I keep thinking of is the disease is a lock, and traditionally what we do is we just it's almost trial and error. We try to find that key that unlocks it, but the ability to know what that structure of that lock actually is, and then and then is customizing the key so it fits that structure. Does that make sense?
Absolutely. It makes sense. It makes perfect sense. And, I think it's something that, like, theoretically we've known is possible as we started to get into technologies like cryo-electron microscopy to be able to understand the structure of proteins, especially in tumors, as we started to understand what we could do with genomic sequencing. Like, these are all these things we've been getting better and better and faster at over the last decade. Theoretically, we knew these things could happen. The biggest hindrance has actually been the development of software to mesh these pieces together to make that doable. And, it's super interesting because we saw some software development over the last 5 or 6 years focused on the small molecule space. Small molecules are so
easy. That's easy.
Yes, it is. It's so simple compared to a biologic. And so they really haven't been able to contribute to the space that in the way we hoped that they could. And that's absolutely an area where we believe that this software can completely disrupt the speed and the safety and the efficacy with which we were approaching personalized medicine. And it can help other companies who simply need a way to harmonize all of that data from their patients, from previous trials and the existing data out there, to be able to analyze it, because they can technically do that with our software on a SaaS model. We can organize that data isolated for them so that we don't have access to that data. And then they can use our software to be able to research all of that information and harmonize that data and make better decisions. And that's definitely an area that we have seen beginning to ramp as we've gotten BioStrand's word out there.
I mean, so the next question actually is sort of tags on to that. It seems like everything BioStrand is almost happening faster and faster. You're gaining speed here. When do you anticipate being able to, see a revenue-based contract, like where you can actually say, okay, you know what, we just made some money with the BioStrand product? Yeah,
I would say in either this quarter we're in or the next quarter. Okay. So next 2 quarters, I think is reasonable. The current program interest that's coming in right now with regard to the use of our Lens AI software for in silico modeling, whether it's humanization or biosimilar, those are all programs that are going directly to BioStrand. And obviously the secret sauce is with BioStrand. So we expect to see that quite rapidly.
Fantastic. Fantastic. Yeah. Okay, so I hopefully I get this question right. It says, can IPA patent this technology? So I'm assuming BioStrand. I mean, we've got patents around BioStrand, or is it open source to copy BioStrand technology? Yeah, I mean, correct me if I'm wrong. BioStrand, there's a number of different patents you have around BioStrand and sort of what it's capable of doing. I imagine there's probably going to be more down the road as you find out really what it's capable of doing. But it's certainly not open source.
No, absolutely not. And I think it would be extremely challenging for any group, no matter how large they are, to replicate this. The patents that have been filed for BioStrand actually have not yet been approved. They filed 3 different patent families, so multiple patents within 3 different families that were in BioClue, BioStrand, and BioKey, the 3 different subsidiaries that we purchased. And all of these patents had been submitted, they had been reviewed, and then one of the things that came about was, again, it's the details around these fingerprints and exactly what they represent and exactly how they've been coded they are really complex. And so there was, a lot of back and forth with reviewers and questions. And then they wanted I don't remember the exact word for it, but they actually wanted like a verbal face-to-face review and questioning of the patent. We weren't of the patents, all of them. We weren't entirely sure what to expect of that, or if that was potentially not a good sign, because it's not good to confuse patent lawyers, but we also need to be very careful about what we put into patents. So that was very interesting, because that now was completed this week, and that was what I referenced in the beginning of this call. Is they actually came back with that they are satisfied with the conversation we had, and the unofficial statement that we received is that they do recommend moving forward for the approval of these patents that have been applied for.
Fantastic. Yeah. Perfect. Okay, good. Okay, so clearly someone that is much smarter than I am, because I barely understand this question. When you speak about structure, are you referring to de novo protein design? If not, what is BioStrand's technology differentiator?
So when I refer to, I've referred to structure in a few different contexts. When I refer to the fingerprints, referencing a sequence or a structure. So we have fingerprints that are sequence fingerprints. And then we have fingerprints that are structuralized versions of the fingerprints. Those structuralized versions of the fingerprints are in silico designed structures where we say this fingerprint actually gives rise to this structure, right? And then it's that structure that's going to dictate the function. And now that we've indexed those structuralized fingerprints, we can also search with structure. When I talk about in nature, the sequence structure function relationship, I'm just referring to any biological molecule. Any DNA sequence is going to give rise to the protein, which will have a very specific structure based on the sequence. And then that structure will dictate what that protein can do. So there's a number of ways where the LensAI software is a differentiator. With regard to the LensAI software and the structure, the main way that it's differentiated is that we have structuralized fingerprints that are unique to BioStrand. They have identified these fingerprints that are common in the structure of a molecule that relate to the sequence and that relate to a function that have been indexed in the software, so that when we put in any information at the sequence level or at the structural level, at even the sequence of a HIFT or a full sequence or massive amounts of data, it doesn't matter. When it goes through and searches, it's going to find the fingerprints at both the sequence level and the structural level. That will relate to any information in the database about similar sequences or similar structure, and that structure will always give us input into a particular function. So at the core of that is definitely the HIFs themselves, the identified and structuralized HIFs. With regard to our ability to build novel in silico proteins that retain structure and function, we actually don't disclose how we do that and what we do within there. But what I can say, what we can do is we can build a novel molecule while focusing on retaining a structure and not on any existing particular sequence.
Fantastic. Hopefully that's the answer they were looking for.
So.
Still, a lot of it was way over my head, which doesn't take much. Okay, this Talem-related question, and the question is: it looks like there hasn't been a new asset out of Talem since August 2021. Is this due to financial allocation, workload capacity, or something else? You guys are continuing to work on sort of early stage discovery antibodies, but you're anticipating anything significant to come out, or maybe.
So we've had a couple new assets actually enter into Talem, and we've had new versions of existing assets. So we do have a very interesting T-cell engager, and we did actually develop a bispecific within Talem that didn't exist previously. and collect all of the in vitro data, functional data with regard to our ability to engage and T cells, and with reference to our ability to fight cancer with this particular molecule. We're in the process right now of actually patenting each of those individual molecules as well as the bispecific product. And then, yes, we've had a couple of other programs that have entered that we aren't really quite as vocal about, as we really focus on these four that we're interested in moving forward and moving out. Now, with regard to that, there are a couple of programs that we did say, all right, we have a lot of programs in here. We really want to make sure that these four have everything they need to get out the door. We really want to make sure that we're building a best in class with this bispecific. We have a couple here that we know have great demand with regard to the market and patient need that we're going to continue to emphasize. There are a couple of other ones that we deprioritized and we slowed down or we've halted for now, in part because we don't going forward, looking forward with regard to with regard to financials, it's not necessarily as based on that as it is with regard to, we want to commercialize a product to get Talem self-sustainable so that the income from Talem is driving all of the product development in Talem, and not get ahead of ourselves before we hit that commercialization point. So where some of the products just really outperformed others with regard to functional data, with regard to interest externally, we wanted to put our emphasis on those particular programs.
Jennifer, speaking of sort of currently the four assets that are likely to be, looking to be licensed, what can investors expect as sort of a ballpark or framework around what you guys want to see in a commercial agreement on them?
Sure. So, with each one of those products, we actually have a spreadsheet. We don't share that with the people we're talking to because it's pretty complicated. We don't want to overwhelm them, but we have a spreadsheet specifically with regard to preclinical products and what stage of preclinical development they've gone through that have happened historically. What did those deals look like? What were the financial ranges around them in terms of upfront payments, milestones, and royalties? We have a very good sense of where the products that we have fall into the commercial space with regard to historical deals that have been made. And, the ranges always, vary, but obviously upfront payments are always in the millions. And then depending on the novelty and the need of the product and the buyer, quite frankly, then, you see kind of some variations in those ranges. But absolutely every one of those structures involves in some form or another, but always together, an upfront payment, milestone payments, and royalty payments. Some of the variations that you might see in that type of deal structure are usually around the milestone payments. For instance, if there hasn't been in vivo functional data, you'll oftentimes have kind of a little bit of a backload on that very first milestone. Milestones in additional preclinical data with an IND application, and then moving through each of the milestone stages. Some of the things that you might sometimes see tweaked or even added to that are, additional payments, not only completing a clinical trial, but in the introduction of a clinical trial, and even more so around royalty payments. So there are oftentimes royalty payments that are kind of tiered, if you will, like it's a certainty royalty percent up to a certain amount of commercial income for the third party buyer. And then that percent might change once you hit a different overall amount of commercial income for that company. So there's a lot of different ways that can be sliced and diced, and that becomes kind of the details of the negotiation with each individual company. But by and large, as a general platform for an agreement, they are upfront payments, milestones, and royalties. And that's, we've not, with any of our current discussions, steered from that general format.
And you mentioned, we're looking at potential upfront payment in the millions. It's not, a couple thousands. It's in the millions of dollars. Okay, perfect.
Exactly.
Yeah,
exactly.
Perfect.
Definitely in the million.
Yeah, awesome. Okay, see some comments here. Well, those are more comments, I guess. why don't we, I mean, we've spent so much of your time here. I think we're getting close to 2 hours here. I mean, is there anything we sort of missed? I mean, is there anything that you want to talk about that we didn't really get a chance to talk about? Is there anything new that you guys are looking forward to? I mean, you mentioned, what is it, function first B cell technology. That's the first time I've heard of that. So clearly you guys you're constantly innovating as well. Is there anything sort of new and exciting that we should all be watching out for?
Well, let's see. I would say, you know, our biggest milestones are ones we've covered here that we're really looking forward to. It's always, you know, it's definitely that Talem commercialization. The BioStrand is really what's had us hopping for the last 8 to 12 weeks. You know, we've all kind of, you know, pivoted a certain amount of our attention to making sure that they have what they need to get these case studies out the door, so that we can really demonstrate with extreme clarity and transparency to our pharma partners exactly what we can do for them. I would definitely look for some of those agreements, you know, again, you know, overall, a number of agreements ranging from, I think a few of them that were negotiated prior to, you know, where we started talking a little bit prior to our bigger findings, are ones where we expect them to be tremendously impactful, but revenue at a slightly later stage. Whereas the newer ones that we're negotiating and we still anticipate, you know, getting through this quarter or next quarter are ones where we would anticipate early upfront money, really, for any sort of even initial wet lab work on those particular programs. So I'd definitely be watching for those. Apart from that, I think that, you know, we're still ramping, as I mentioned, in the contract research work. Our pipeline is growing faster than ever. The amount of work starts, especially at our Canadian site, is up tremendously. I think that is by and large due to our function first B cell, where, you know, competitors are oftentimes just giving out a lot of genetic information about potential antibodies, but we're giving out very rapidly information about exactly which B cells producing an antibody with a specific function, and able to also turn around then the information on binding function and sequence within an extremely rapid amount of time from first sorting those B cells. And that's just gaining a ton of interest because people don't have to go through thousands of sequences or hundreds of antibodies. They can go right to the ones that are getting down to business and doing what they need them to do. And well, we know of no other platform that comes close to being able to do that. And that's driving a lot of that growth in Canada. So we're very excited about that. Hiring it for sales teams has always just been a challenge since the second half of COVID here. We actually just landed a wonderful salesperson who will be starting with us toward the end of October, which as our most of our great hires during COVID are coming through people that we know well, people that we've been close to in the industry. So we're very excited about that growth and that's continuing to be able to get the word out there. Apart from that, we're looking forward to having our AGM in person again this year. We're really hopeful. We're making backup plans because we have this kind of dichotomy of things going on where we're also, you know, quite sure we need to be prepared for what may happen with different variants. So so we are planning to do that, have our AGM in person up there in Canada. So we'll get out information on that as well as backup plans in the event that COVID takes precedent. And yeah, apart from that, we look forward to sharing information in the near future here about these deals that are arising and these different ways that we're formulating new ways for people to access IPA's technology within BioStrand and new types of deal structures for IPA, which is really exciting for us. And in the meantime, we're really also, you know, just ramping those downstream capabilities at IPA, such as our protein production and characterization, so that we can rapidly support the in silico products coming on to move them as quickly as possible toward the clinic. So,
so lots on the go.
Lots on the go.
Just as we started the conversation, you've always been busy, and you certainly haven't slowed down here. Yeah, I mean, clearly it sounds like there's a lot of things that are sort of, you know, getting closer and closer. exciting stuff in the BioStrand. The big takeaway I got today really is that I think we're much closer to commercialization of BioStrand than I thought.
Yeah.
So that's fantastic. Telum continues to do what it does best, and Polytope, fingers crossed we can get it into the clinic and have a big impact, on humanity, quite frankly.
Absolutely.
Well, this is fantastic, Jennifer. I mean, we've touched on so many things. Is there any key takeaways? Is there any key message you want to make sure that everybody walks away with today that you feel is important?
I would say the biggest take-home message from IPA is that we really truly believe that the way we are going about changing how algorithms are used, and changing how we look at how we can very, very rapidly build safe and effective drugs is really a disruption. We anticipate this being a full disruption in the market, not just of in silico antibody discovery and design, but the amount of money that people need to pay and the amount of time it takes to do things like transgenic animals and humanization. And the way that we can go about using existing information out there in the literature, in patents, and in drugs that are commercialized to make better drugs and make them faster and not be thwarted by the patent landscape. And that's all about getting better drugs to patients faster and overall cheaper for biopharma, because we're doing it in a way that eliminates a lot of the discovery risk in the laboratory. And so from our perspective, the most important thing that we were looking for when we went out to look for an AI and an ML component was market disruption, not just market and discovery and development of antibodies, but market disruption in algorithms and how we use AI and how people think about how AI can transform healthcare and agriculture. And, we loved what we saw at BioStrand compared to other companies, and we're just more confident than ever that what they have demonstrated and what they're demonstrating now has brought us exactly what we had hoped we had. It just brought it to us faster than we thought we would see it. And I think that's the take home. It's about, for us, it's about market disruption. It's about making this a better way to produce safe and effective drugs. And so I would really say that, watch for our updates, watch for information on those different partners that we've been able to come in as early adopters. And certainly don't take your eye off the ball on Polytope and Talum. Those are there and those are still strong, and that CRO is still growing, but it's just, it's all coming together for us to be a part of a bigger, better company that's really starting to take shape and take on its real, I think, personality and identity in a way that is just clearer and clearer, both internally and externally.
Fantastic. Jennifer, thank you so much. I can personally attest to the fact that you're one of the hardest working people I know, certainly one of the hardest working CEOs I've ever worked with. So taking 2 hours out of your time is extremely valuable. So we really appreciate that. Clearly, we're big fans of what's going on. I certainly am, and we'll continue to cheer on from the sidelines here and hope for the best. So again, thank you for taking the time with us. We've been speaking with Dr. Jennifer Bath, CEO of Immunoprecise, symbols IPA, both on the Venture Exchange and on NASDAQ. And Jennifer, we look forward to catching up with you as soon as we can again.
Thank you so much, Paul. It's great to see you.
Thanks.
Thanks. Take care, everyone.